chapter 12 from your Medal textbook. Okay, the outline. Okay, dalam topik yang ni, what covers in your syllabus is just 12.2, 12.3, 12.4 and 12.5. However, in order for you to understand what happened in 12.2, I will go back and just recap on what is the structure of DNA. So maksudnya saya akan masuk sikit lah on 12.1, but it's not included in your syllabus. Cuma dia refresh balik lah apa yang you belajar masa SPM dulu ke, atau what your basic knowledge on DNA is like. Okay. Alright, so the learning outcome. By the end of this topic, which is topic 4, you have to be able to describe the enzyme and proteins involved in DNA replication, and also to explain the function of transcription and translation. Okay. Okay, so before kita start talking about replication of DNA and everything, so we go back to the basic on what is DNA and why we call it as the genetic material. So DNA and proteins are candidates for hereditary material. Okay, it's passed on from generation to generation to generation. So what is the requirement of a genetic material? There are 3 requirements that is good for your knowledge sebenarnya. They must be able to store genetic information. Baru dikatakan dia sebagai genetic material. They're able to store the genetic coding or the genetic information. Lagi satu, next point, it must be stable and able to be replicated accurately during cell division and transmitted from generation to generation. It must be stable. Jangan ada yang tak stable and then which akan contribute to non-disjunction and everything. Kalau boleh, the requirement for good genetic material, it must be stable. Okay. Another one is must be able to undergo mutation to provide genetic variability. These are the 3 main points for the requirement of a genetic material. Okay, so we go back to the basic for structure of DNA. So if you refer to page 211, you'll be able to see the nucleotide composition of DNA. So DNA contains 2 nucleotide with purine base. Purine base ni is a double ring. Okay, 2 with purine bases, which is adenine and guanine, which have a double ring. Okay, purine. Contoh adenine and guanine. Lagi 2 nucleotide is pyrimidine base. Pyrimidine base maksudnya dia ada single ring only. Kalau single ring ni is thymine and also cytosine. Okay, selalunya masa pairing, complementary pairing of the bases, adenine will pair up with thymine. Okay, purine dia akan connect dengan pyrimidine base. So adenine will pair up with thymine, A with T. Guanine pairs up with cytosine. So G is with C. A, T, G and C. Okay, you have to remember that. So this is the basic structure of DNA. So DNA uses complementary base pairing to store genetic information. Okay, there's one rule of Chargaff. Nama dia Erwin Chargaff. Dia mengatakan bahawa the amount of the DNA dalam A, T, G and C dalam DNA itu are constant among members of the same species, but vary from species to species. So in each species, yang pentingnya apa yang dicakap Chargaff's rule ni, there are equal amounts of A and T and also G and C. Maksudnya if you see here for Homo sapien, which is human, kita lah kan, if you see the amount of DNA composition dalam A is 31%, dalam T is 31.5. So if you see these two are similar, so we can say they are equal amounts of DNA composition. Kalau G and C is 19.1 and 18.4, dia tak lari jauh. So we still can consider it as equal amounts of DNA composition. So if you tengok next is the Drosophila. the fruit fly. Kita tengok dia punya A and T also lebih kurang sama. And then G and C also lebih kurang sama. So the same goes for all the others. Corn, fungus, bacteria. A and T punya DNA composition is equal amount. And G and C are of another equal amount. Okay, this is the Shagaf's rule. You have to remember Shagaf's rule. Dia mengatakan bahawa A and T, the amount of A, T, G and C in the DNA are equal for A and T and also G and C. Okay. So structure of DNA. So this model was introduced by James Watson and Francis Crick. Kita akan selalu dengar Watson and Crick model of DNA structure. So what actually is the model of DNA ni? So they came up with the model tu, which they stated that it is a double helix model in the shape similar to a twisted ladder. Okay, kalau you tengok on page 213, you can see dia macam tangga sikit, but a twisted ladder. Okay, and then they have a sugar phosphate backbone make up the site, which is this. Okay, this is the phosphate deoxyribose backbone. And then they have the hydrogen bonded bases make up the rungs. Okay, hydrogen bases. I think these are all hydrogen bases. Tapi yang dalam ni tak berapa nak nampak dia punya dot tu. Okay, but they have a hydrogen bonded bases which makes up the rung. And then the two DNA strands are anti-parallel. What does it mean by anti-parallel? Dia tak sama direction. So this one is 5 prime, this one is 3 prime. This way is the direction. Tiba-tiba this side is this way, 3 prime to 5 prime. So it is, the two strands are anti-parallel. Okay, so information stored in the DNA must be read in the 5 prime to 3 prime direction. This is very important. DNA must be read in the 5 prime to 3 prime direction. So DNA is replicated in a 5 prime to 3 prime direction. Okay, jangan kata 3 prime to 5 prime. Kalau 3 prime to 5 prime, you will need Okazaki fragment. So this one is 5 prime to 3 prime. So complementary base pairing ensures that a purine is always bonded to a pyrimidine. Purine kena bonded with a pyrimidine. A and T, G with C. Okay. So this is the model. You can see this on page 213. So these are the two pairs, Watson and Crick. Okay. Okay, so next we go on into the real one, which is the what covers in your syllabus, 12.2. So 12.2 is the replication of DNA. So when does DNA replication happen? Anybody can answer that?
During mitosis in mitosis.
S phase.
Cell division. Sebenarnya kan masa yang S itu betul. So DNA replication happens before cell division. Dia kena replicate dulu baru dia akan divide. Okay. So it is DNA replication is called semi-conservative. Semi-conservative, what does it mean? Dia mesti ada satu yang lama, satu yang baru. Okay. Each strand of the original double helix serve as a template for a new strand in a daughter molecule. Okay, dia mesti ada satu lama dengan satu baru. Then you will call it as the semi-conservative replication. So DNA can only be synthesized in a 5 prime to 3 prime direction. Okay, remember this, 5 prime to 3 prime direction. There are 3 steps of DNA replication, which is unwinding, complementary base pairing, and the last one is joining. So we will go into detail on this. Okay, sebelum kita tengok video on how DNA replication is like, so kita akan go through what are the proteins involved in DNA replication. So the first one we have the DNA helicase. DNA helicase yang dekat sini. DNA helicase. Okay. This separates the double-stranded DNA into single strand. Dia buka kan. Okay, this is the DNA helicase. Next one is the single-stranded binding protein. We call it as the SSB. So you see here, kat sini. This binds to single-stranded DNA and prevents it from reforming a double helix. Dia kan bila dah DNA helicase separate the double-stranded tu, ni yang maintain dia. Maksudnya dia maintain it to be single-stranded. Dia prevent it from reforming balik, joining balik jadi macam double helix balik. So next one is DNA primers. DNA primers ni kalau you tengok YouTube, banyak yang tak mention, tapi dalam syllabus kita ada DNA primers. Okay, so DNA primers is synthesizes short RNA primer. Dekat sini dia for the start. Mana dekat sini ya? Oh, tak ada pula dekat sini. Oh, here. This is the DNA primers. Okay, and then next is DNA polymerase. The big one here. This is DNA polymerase dekat leading strand. This is called the leading strand, and di kat bawah ni is called the lagging strand. Lagging strand is 3' to 5'. Kalau leading strand yang biasa 5' to 3'. Okay. So DNA polymerase, what does it do? It synthesizes DNA in the leading and lagging strand. Kerja dia memang synthesize DNA. So dia ada dekat leading strand, dia also ada dekat lagging strand. Okay, it removes the RNA primers, filling the gaps with more DNA. Okay, bila dah ada DNA primers ni and then RNA primer kat sini, dia akan remove, dia akan fill the gap with more DNA. Nanti dalam video you will be able to see it. Okay, it also proofread newly made DNA. So the last protein is DNA ligase. Is this one. Kalau ligase ni you kena ingat dia macam glue. Kerja dia macam glue, Yuhu glue lah kira. Dia attach dua Okazaki fragment, which is adjacent Okazaki fragment in the lagging strand. Okay, beza leading strand dengan lagging strand. Kenapa? Apa perbezaan dia? Does anyone know? Leading strand ni macam mana? Lagging strand ni macam mana? Ada tak? Okay, leading strand, if you remember I said it is 5' to 3'. Kalau lagging strand pula, leading strand kan tadi. Okay, lagging strand is 3' to 5'. Tapi the DNA punya direction for synthesizing of the DNA is always 5' to 3' sahaja. Because this side is 3' to 5'. That's why dia kena do it in stages or in fragments. We call it as the Okazaki fragment. Okazaki fragment ni hanya ada dekat lagging strand. Dekat leading strand tak ada because it's normal. Okay, dia boleh synthesize normally. Okay, nanti kalau you tengok video tu, I think you akan faham lagi. Okay, let's go into the video on DNA replication.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates the strands of the DNA molecule, which are stabilized by proteins. The enzyme DNA polymerase initiates the process of replication by adding nucleotides to a pre-existing strand of DNA. However, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add complementary nucleotides to the growing strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. one strand is replicated continuously towards the unwinding, separating portion of the original DNA molecule, while the other strand is replicating discontinuously in the opposite direction, with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Okay, so if you see in the video tadi, there are 3 steps involved. So the first one is the unwinding. Lepas inilah terjadinya formation of the replication fork. Kalau you tengok sini, replication fork is V. Asal V je, we call it as the replication fork. Okay, this is the replication fork. Dia nampak bentuk V and the helicase will be here. So awal-awal, the separation of these 2 strands of the parental DNA molecule is by the enzyme DNA helicase. Okay, bila dia dah bukakan, so the SSB akan tolong attach to the newly separated DNA which prevents the helix daripada jadi double stranded balik. Okay, so that's the job of SSB, which is single stranded binding protein. So dalam first step unwinding ni is actually the formation of the replication fork. Yang terlibat, enzim yang terlibat adalah DNA helicase and also single stranded binding protein, which is SSB. Okay, the second step. Okay, second step is the main step. Bukan nak cakap the main step lah, but most of the apa ni? Function, most of the apa nak cakap ni? Most of the event happen here, dekat sini, dekat complementary base pairing. Okay, so what does it mean by complementary base pairing? It is between a new nucleotide and a nucleotide on the template strand. Okay, this is called semi-conservative replication. New nucleotide and a nucleotide on the template strand. Okay, so DNA primers. DNA primers ni places short RNA primer on the strand to be replicated. Macam you nampak dalam video tadi, dia letakkan the RNA primer. Okay, lepas tu baru the DNA polymerase recognize the primer and begin the DNA synthesis. Okay, dia macam mana dia buat DNA synthesis ni is by adding new nucleotide complement to the old nucleotide. Okay, they connect new nucleotide in a chain. Maksudnya daripada tadi dia recognize the old strand, bukan the old strand, the template strand, and then they add new nucleotides onto the strand. So there are 2 strands that are replicated differently. The first one is the leading strand yang you nampak continuous tadi kan. The continuous one is the leading strand. While the lagging strand is segmented replication, also known as Okazaki fragment. Dia in fragments, lepas tu dia DNA polymerase akan synthesizekan, tapi in fragments. So last kali DNA ligase yang akan glue them together. Okay, so this is the second step, complementary base pairing. So last one, as I said, is the joining. So joining ni, joining of nucleotide in the lagging strand by DNA ligase. Okay, joining ni dalam lagging strand. Yang lagging strand yang bahagian bawah. It can be atas juga, but you have to know that it is in the lagging strand. Lagging strand is by the enzyme DNA ligase to form the new strand. So each daughter DNA molecules contain 1 old and 1 new strand. So this is called semi-conservative replication. Okay. So let's watch another video. Yang ni video daripada YouTube yang saya dapat, but I think this one is dia menarik sikit dia punya compared to the McGraw Hill ni video.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand.
Okay, so I think that video was very detailed. So kalau you tak faham bahagian DNA replication, please watch the video. Sebab saya rasa video tu sangat membantu. Okay? So next we go on into prokaryotic versus eukaryotic replication. So yang ni dalam prokaryot. Prokaryot sebab dia bulat. Okay, dia ada single circular loop of DNA. Dia bulat macam ni. So macam mana dia replicate? So how does a prokaryot replicate is by it move in both direction circularly. Maksudnya nampak ni jadi dua. Jadi the replication moves around the circular DNA molecule in both direction. So it produces two identical circle. Okay, nampak ni dia move dua. Replication occurring in two directions. Satu sini dia boleh, lagi satu sini pun dia boleh. Satu sini, satu sini. Dua-dua dia boleh. So that's why dia boleh jadi cepat. Replication in prokaryotic is quite fast. It takes about 40 minutes. But the cell divides every 20 minutes. Okay. So the process begins at the origin of the replication. This is the origin. So a new round of replication can begin before the previous round is completed. It doesn't mean that dia kena tunggu 40 minit habis baru another replication boleh happen. No, it can happen simultaneously juga. It doesn't have to wait for the first round to finish baru second round yang replicate. Okay, so this is prokaryotic. It's a bit different than the eukaryotic. Prokaryotic you kena ingat dia bulat. So dia dua direction. Carrying in two direction but bulat. Okay, so in eukaryotic, okay, eukaryotic kita tak bulat. So dia ada replication bubble. which spread bidirectionally until they meet. Nampak ni, this is the replication bubble. So from here, dia akan kembang new strands kan. Lama-lama dia akan connect sampai jadi a long new strand. Okay, so this replication is semi-conservative and polymerase is unable to replicate dekat the end of the telomere. So ini kalau kat DNA kita ada telomere, dia ada protective tip. So dekat sini tak ada coding apa-apa dah lah. So dia akan stop sampai sini saja. Okay, so kalau yang tadi dalam prokaryote, tengok dia tak ada telomeres, dia bulat je sampai habis. Okay, kalau dalam eukaryotes, dia ada ending dekat sini. Dia macam panggil cap protective tip. So dia dah stop sampai sini. Okay, so that is the difference between prokaryotic and eukaryotic replication. So what is the accuracy of the replication? Selalunya DNA polymerase is very accurate. However, it can still make mistake, which is about once for every 100,000 base pair. DNA polymerase ni, they capable of identifying and also correcting error. It is sangat-sangat, how do you say, very low percentage to make a mistake pun. It's only once per 100,000 base pair. Okay. So next subtopic, we will go on into the genetic code of life. Ataupun dalam buku you, it says gene expression, RNA and the genetic code on page 216. Okay, so this is more of a gene expression. We deal dengan RNA untuk subtopik ni. So there's a mechanism of gene expression. Okay, DNA in gene specify the information, tapi the information is not structure and function. Selalunya yang deal dengan structure and function is gene expression. Bila dia dah express through protein synthesis, RNA, okay. DNA, RNA, and then jadi protein synthesis. So this part is the structure and function. Kalau DNA saja, it only specifies the information. Okay, the expression of genetic information into structure and function. So DNA in a gene determines the sequence of nucleotide in an RNA molecule. So bila RNA molecule ni controls the primary structure of a protein. Bila ada protein ni, RNA is translated to a protein, baru terjadinya gene expression. Baru the coding untuk specific type of the folding of the protein lepas tu. So this is called the central dogma of molecular biology, which is the flow of information from DNA to RNA and to protein. Okay, to an observed trait. So protein ni carries a certain trait. So protein ada banyak, there's a lot of function of proteins. Okay. Okay, so the central dogma of molecular biology again, dia melibatkan dua step yang important, which is transcription and also translation. Okay, from DNA, it is transcripted in the nucleus jadi mRNA. So mRNA ni bila dia translate the ribosome, dia akan jadi protein or called the polypeptide. Polypeptide ni depends on the codon and dia akan code for a certain function. Okay, this is amino acid. Okay, so transcription process by which an RNA molecule is produced based on a DNA template. So that is transcription. Kalau translation, it is the process by which the mRNA transcript is read by a ribosome and then converted into sequence of amino acid in a polypeptide. Okay. So we will cover transcription and translation in the next lecture in detail lagi lah. This is just a brief introduction on transcription and translation. Okay, so next we go on into RNA. So RNA, what is the difference between RNA and DNA? RNA is ribonucleic acid. It is a polymer of RNA nucleotide. So RNA nucleotides contain sugar ribose. Kalau DNA is deoxyribose. Okay, this is DNA. So this is RNA. Okay, dia ada sugar ribose, bukan deoxyribose. So RNA nucleotides are of 4 juga, sama, cuma bezanya dia ada uracil, dia ada adenine, dia ada cytosine, dia ada guanine. Dia tak ada thymine. dia tak ada thymine, dia ada uracil. So this uracil U replaces thymine, which is the T of the DNA. Kalau RNA, you must remember tak ada T, dia tak ada thymine. Dia only ada U, which is uracil. Okay. Okay, type of RNA. Dia ada 3 that we have to know. The first one is mRNA. It takes a message from DNA in the nucleus to ribosome in the cytoplasm. So this is messenger RNA or mRNA. This is ribosomal RNA, which is rRNA. This is along the ribosomal protein which makes up the ribosome, where polypeptides are synthesized. Okay, nanti in the next lecture you will be able to see the function of the rRNA. Okay, last one is the transfer tRNA. So this is tRNA that transfers the appropriate amino acid to the ribosome. Okay, you must remember RNA ada 3, which is messenger RNA, ribosomal RNA, and transfer RNA. mRNA, rRNA, and tRNA. Okay, what are the main difference between RNA versus the DNA? So kalau you tengok sini, apa the main difference dia? Structure dia ni. This is RNA and this is DNA. What can you see?
Dia bukan double
Helix.
helix.
Ya,
Helix
betul.
dia ada
RNA
satu.
is just single. Okay, RNA is single stranded and it is not in the helix punya shape. Okay, this DNA is double stranded and yes, it is in a helix shape. Okay, so bases dia, what you have to know, RNA ada 4, which is C, G, A, and U. Kalau DNA ada C, G, A, and T. Okay, and then the sugar phosphate group backbone dia tu, sugar dia, kalau RNA is ribose. Kalau DNA is deoxyribose. Okay, that is the main difference between RNA and DNA. Okay, next we go on into the genetic code. So genetic code is usually called codons. I'm sure you heard of codon before. Okay, these are codons. Dia selalunya 3. Okay, satu set ni one codon. Satu set lagi a different codon. Ni pun another codon. Okay, there are 20 amino acids found in protein which is uniquely specified by one or more codon. So this codon, katakanlah A, G, C ni, it translate into a serine. Kalau codon 2, which is GAC, it translate into an aspartate. Codon 3, CCC, contohnya ni, this is a proline. Okay, so they use codon is symbol. The symbol used by the genetic code are mRNA bases. So they function as letters of the genetic alphabet. So genetic alphabet ni kalau dalam RNA ada 4 je, which is U, A, C, and G. Okay, so codons in the genetic code are all 3 bases. 3 bases long. They function as words of the genetic information. So this is a letter contohnya, and this is the word. Okay, so there are 64 possible arrangements of 4 symbols taken 3 at a time. Okay, daripada 4 ni, there can be ada 64 possible arrangement of 4 symbols taken 3 at a time. Codon ada 3. So they often referred to as triplet. So selalunya genetic language ni only has 64 words. Okay, jom kita tengok in detail what are the codons. Okay, codon untuk stop and go, untuk nak mula-mula start translation, dia akan start dengan AUG. AUG. Okay, codon AUG. Codon AUG untuk start AUG. Kalau codon stop, I always tell my student UA, UA, UGA. Always remember that. Macam, okay, UAA, UAG, and UGA. Kalau dia nampak yang ni, any of these 3 codon, dia akan stop replication. Or stop synthesizing, sorry. So UAA, UAG, and UGA. UA, UAG, UGA. For stop. For starting, it is AUG. AUG. Okay. So these are the possible combination of the bases. Okay, dia boleh jadi macam ni. Dia ada 64 semuanya. But what you don't have to remember every single one. What you have to remember is just the start and the stop codon. AUG for start, UA, UA, UGA for stop. Okay. Okay, so these are the amino acids. So yang ni you don't really have to know selalunya. Okay, this is just for your own knowledge. Okay. Okay. So what are the properties of the codon, the genetic code ni? It has to be universal. Okay, with a few exception. All organisms use the code the same way. So they encode the same 20 amino acids with the same 64 triplets or 64 codon tadi. Ada 3 dan 1 kan? And then it should be able to degenerate, which is it will be in abundant. Okay, it can be in abundant, it can be in redundant as well. So there are 64 codons available for 20 amino acids. So most amino acids are encoded by 2 or more codons. Selalunya, it's about 2 or 3 lah, 2 or more codon. So it is unambiguous, which is it is exclusive. Maksudnya dia each of the codon tu ada meaning sendiri. Maksudnya tadi AUG tu meaning apa? It is a start codon, and UA, UGA is the stop codon. It has a meaning for each of the triplet codon. So each codon specifies only one of the 20 amino acids. It contains the start and the stop signal. It's like the punctuation codon. Like the capital letter we use to signify the beginning of a sentence and the period to signify the end. Okay, that is the genetic code. Dia mesti ada 4 ni. It has to be universal. It has to be, can be generated, degenerate. It can be unambiguous. It can be contain start and stop signal. Okay, so this is the last video for this class. Just an overall view of how DNA and protein synthesis is like. Okay.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so